we're going to diagram cell division. There are a few objects to keep track of here. We'll start with our cell that will represent prophase. A cell in prophase is going to have a nucleus that has the nuclear membrane disappearing. And inside of that nucleus, you should have replicated DNA. So all your chromosomes should indeed look like X's. We're going to do 4 X's here for good measure. So 4 chromosomes that have been replicated. In addition to that, we're going to need 2 sets of centrioles. And those are going to be sending out these spindle microtubules. The centrioles and the spindle microtubules together form this object that we collectively call the spindle. That's prophase. For metaphase, it should be a cell that is not terribly elongated, about the same shape as the other one, except now the spindle, which we can draw again here relatively quickly, has done a rather excellent job of moving all of our chromosomes into the exact equator of the cell, preparing to split the sister chromatids apart from one another. So what we should see are some of our spindle microtubules connecting directly to the middle of those chromosomes, right at the centromere, and other spindle microtubules that run past the length of the cell. Some of these pull, some of them push. We also need a cell that is in anaphase, and so this cell should be a little bit elongated, almost like a long peanut. And what this is going to show you is, once again, the centrioles have begun to pull our little chromosomes, which are now called chromosomes, even though a minute ago they were just sister chromatids. They have left their identical sister chromatid pair and are now being pulled towards opposite ends of the cell. And again, the objects that are pulling this would be these spindle microtubules coming out of these centrioles. In addition, we should have some that are helping to elongate the cell, just for accuracy. And then finally, what we have in telophase, you should have basically what looks like 2 cells that are right next to each other and are not quite completely finished yet. And so what you'll have here is a spindle microtubule that is starting to dissolve. Yes, the spindle is going away, one on each side. And what you're also going to have are your 4 sister chromatids that are now in their own little area. The nuclear membrane is starting to reform, so this is eventually going to be the nucleus. And in addition, you could label or note the cleavage furrow or the place where cytokinesis is occurring. And that's the real important information for cell division, shown here with a little bit more labeling and notation of what's going on.